Good afternoon, students. Here we are going to discuss in detail the production of BCG vaccine, that is a live attenuated vaccine. Now, BCG vaccine is a conventional vaccine, a live attenuated vaccine, where the abbreviation BCG stands for Bacilli of Calmette-Guerin. Now, this vaccine is used for immunization against infection caused by Mycobacterium tuberculosis. A preparation called BCG. Okay, we use a preparation called BCG for immunization against infection caused by Mycobacterium tuberculosis. Now, tubercle bacilli was first time isolated in pure culture by Robert Koch in the year 1882. The route of infection of mycobacteria is either by inhalation or by injection. Basically, tuberculosis is a respiratory tract infection, but in severe cases, the disease may spread throughout the entire body. See, actually, now there are various types of tuberculosis which may involve various organs of the body, okay, like brain or bones or eyes and so on, okay. So, tuberculosis of various organs is there, right? The pulmonary infection in man, that is pulmonary tuberculosis, is caused by human type of Mycobacterium tuberculosis. Now, Guerin and Calmette introduced a live attenuated vaccine BCG using a bovine strain to immunize against TB. He attenuated the culture using glycerol bile potato medium. That means severe subcultures into medium. Cereal subcultures, sorry. So he attenuated the culture using glycerol bile potato medium, and the bovine strain is now used for production of vaccines for immunization purpose. Okay, so let us discuss the production of BCG vaccine. What is the organism used? Mycobacterium tuberculosis, strain number 1173P2, that is obtained from Pasteur Institute and is generally used for production of BCG vaccine. What are the culture media that are used for cultivating the organism? The culture is first maintained on LJ medium, that is Lowenstein-Jensen medium, and then it is further subcultured in Dubos medium and once in Engler et al. Medium. Now, Dubos for the first time prepared a homogeneous medium for cultivation of Mycobacterium tuberculosis. The medium contains a dispersing agent twin 80. Chemically, it is sorbitol monooleate. The medium also contains bovine albumin, salts, minerals, glucose, asparagine, everything which is required for flourishing growth of mycobacteria. Later on, Engler et al. Slightly modified the medium for better results. Now, how do we prepare the seed culture, the culture of Mycobacterium tuberculosis? Because for vaccine production, we actually require, the vaccine is actually nothing but preparation of the microorganism. But the organism is in live attenuated form. So we have to grow the culture on a large scale. So we first need the seed culture. The method for production of seed culture consists of 3 subculturing of the organism in LJ medium. Then we subculture the medium for organism for 3 times in Dubos medium and once in Engler et al. Medium. Such several number of subculturing of the organism is required for attenuation of the organism so that its virulence, its pathogenicity is destroyed. Now let us discuss the methodology. A suitable sterilized growth medium like Engler medium or Sortens medium is seeded with definite volume of seed culture. Okay, so once you have seed culture ready, for preparing seed culture, what we do is 3 times subculturing in LJ medium, 3 times in Dubos, followed by 1 in Engler. And for large scale production, we go for suitable mediums like Engler medium or Sortens medium. You inoculate it with definite volume of seed culture. Usually, we've studied all these 20% and 5 to 20% of seed culture. Then it is incubated for a period of 11 to 14 days. Earlier, incubation period was 19 days, but then it was found that given the number of days of incubation are reduced to 11 to 14, then the potency of the vaccine is increased. So now the incubation period of the organism is 11 to 14 days. During cultivation, we have to take care that vigorous agitation of culture medium is detrimental for the growth of organism. So we have to maintain the agitation rate to 200 RPM rate. Okay, it provides good aeration as well as healthy growth of microorganisms. Rate of agitation is 200 RPM. Then aeration rate is 3 liters per minute, which is optimum for the growth of mycobacteria. pH of the medium is maintained in the beginning at 6.8, but as the fermentation proceeds, the pH changes to 7.8, automatically rises. Similarly, we have to maintain the temperature because it's a human pathogen. We have to maintain the temperature at 37 degrees Celsius, and it is found to be most suitable for the growth of organism. Then once the culture of the organism is ready, we have to harvest the culture. Okay, how the harvesting is done? At the end of incubation period, that means at the end of 11 to 14 days, the growth of the organism, that is the culture, is harvested by centrifugation, and bacterial mass obtained is semi-dried. So the whole broth culture is first centrifuged and the liquid supernatant is discarded, and what we get at the bottom is the bacterial mass. This bacterial pellet or mass is semi-dried. The semi-solid and semi-dried bacterial growth is then broken down into fine granules. Okay, by rotating the semi-dried, semi-solid bacterial culture in a vessel with stainless steel balls. Okay, so there is a vessel, rotating vessel. There are stainless steel balls in it. So the semi-dried, semi-solid bacterial growth is transferred into the vessel. It is rotated along with the steel balls. So the you can say culture is broken down into fine granules. Now, during harvesting, when we are harvesting the culture by centrifugation, we add glucose, protein source, and twinating. Why? So as to remove maximum bacterial mass. Okay. So actually the bacterial growth or the bacterial culture is harvested by centrifugation, and during that we add glucose, protein, twinating so that we get maximum bacterial mass. Now, once you have rotated it in the vessel with stainless steel balls, your powder is ready. Then the powder is subjected to dilution of various potency as per requirement, or as per the specifications or requirement, we dilute it to a desired potency. Standardize them and then distribute it into small vials or ampules. These vials or ampules, which contain the powdered form of Mycobacterium tuberculosis, are subjected to preliminary freezing. Okay, and then lyophilization. Lyophilized ampules or vials are again dried over phosphorus pentoxide. Okay, once the suspension is thoroughly dried, the ampules or vials are aseptically sealed. All these procedures are carried out with great precaution and care so as to maintain sterility throughout the production unit. So sterility, maintaining sterility is very important because there should not be any contamination in the vaccine during the vaccine production. Once the ampules are ready, they are sent for control testing. After acquiring satisfactory results, the ampules are labeled. Okay, they are packaged and then labeled. Packaging and labeling. Ampules are packed and labeled under again aseptic condition, and then they can be stored at low temperature. Now the label that is provided on each ampule, each vial, should indicate or should give certain indications as what is the name of the vaccine that is present in the vial, what is the abbreviation. Say for example, it is BCG vaccine, so BCG. What is the date of manufacturing? What is the date of expiry? How to store the vaccine, that means condition of storage, like the vaccine is light sensitive, store, keep away from light and all. So all necessary conditions required for storage are to be explained on the label. And if the vaccine is present in dry powder form, then how much quantity of liquid is injected into patient must be written on the label. That means how much quantity of sterile distilled water is required for reconstitution should be indicated. The label should also provide information about the shelf life of the vaccine, that means expiry date and all. And once the ampules are open, they should be discarded if not used in a particular, if not used at that time. So all these indications are to be explained or put on the label. Now, what are the various tests that are performed for BCG vaccine before sending it into market? Once the vaccine is ready, various pharmacological and pathological tests are carried out in laboratory on animal as well as finally on human volunteers. What are the tests? The toxicity, whether the vaccine is toxin or toxic, that means whether live organisms are present. Virulence, that means because it's a live attenuated vaccine, organism is present in live form, but it should not be virulent. It should not cause any disease. So we have to do virulence testing. Organism should not be living. Okay, it should be, it should be attenuated. It should be live, but not able to cause disease. Virulent. It should not be pathogenic. It should only behave as an antigen. So virulence testing is done. Sterility testing, skin testing, that is tuberculin testing, is also done so that the vaccine, once it is made sure that the vaccine is safe, it is not toxic, virulence has been destroyed properly, it is sterile, and then only it is sent to market for human use. So that is all about BCG vaccine production.